very few summarize in my point of view. What do you mean by transcriptome analysis? So here is a simple view. I mean transcriptome analysis. So first, you see, sure, we should study gene expression. So we have some differential expression, co-expression network, or recently we saw causal network, and there are specific expression, population-based and pedigree-based. But however, this is not enough to study transcriptomics. We also need to study, you see, EQTL, how genetic variation influences the gene expression. And also we should study how epigenomic some variation, for example, methylation and RNA-Seq other, they are not off here. Other genetic element and the chip and histone modification, how to influence the gene expression. And here, why at there, I will say how expression change the signal in the image. So we found that most of the people we study identify some genes with differential expression. However, we can show that even these genes are not differentially expressed, but they will change the morphology of the cell. And they change the morphology of the cell, the shape of the cell. This signal we can observe in the image. So today in my topic will show this one. However, you see, I am a statistician. I should present some methods how to analyze, you see, RNA-Seq data and the image data. So here, I just give you some picture. You see, this is DDA image, the brain. And then we see this brain, if we get segmented into some brain region, you see. This region also correlated if we can form network. And then this is the genes. This gene can be associated or mapped into different subregion of brain. This is the one example here. Another example, this is the cancer, okay, CT. This is also CT can be segmented into several subregions. And this one is tumor size. This one is tumor size. And then this is some genes. And this gene themselves form network. And they also will be associated with this tumor size. And now this is the pathology image. This is normal cell. This is tumor cell. So now we want to study how gene expression will change the shape. This signal here, this is normal, and then this tumor, the shape will be changed. Another question is, you see, we are studying RNA-Seq data. So previously we said one gene expression, we just give one summarized statistic overall expression. However, you see from data, in fact, we observed gene expression, the variation from genomic position to genomic position, they're not the same. How to cause? Maybe due to sequencing technology variation. This is short reads. Previously, someone gave a talk, Kausplin, he said with long reads. Maybe we can eliminate some variation. However, also this variation may come from biology. The data we observed is the curve, not just the one expression and one number. The current so there are two here, now today I will address two issues. One issue, how to extract the signal from image. Second issue, how to get the signal from RNA-Seq data and how to collect them. So the current methods for image genetic data analysis, we can group three. One, just a single variable regression analysis. Second one is multivariate analysis. Third one, chronic correlation analysis. For this one, the whole image just be summarized by one number, maybe average volume, or so here some summary statistics. They are not awful, but however maybe usually we use the simple statistical, just the average signal in pixel or voxel. For the multivariate analysis, which we can use, for example, principal component analysis, and independent component analysis, and clustering, and multi-factor dimensional reduction. For the chronic correlation analysis, we segmented the image into several subregions, and then we can do chronic correlation analysis. This is for image. And then for the RNA-Seq data, most of the current method is just take one gene expression, one number. So however, you see, these methods have some limitation. So image data reduction method do not consider image signal space variation. For the RNA-Seq data analysis, most degree genomic position level variation and real specific variation. So today, my talk will overcome these two limitations. So the basic procedure for image RNA-Seq data analysis. First, we should image feature extraction or selection. They're not of, but today I will present function principal component. Second one, how to get a signal from RNA-Seq data. I take each RNA-Seq data as a curve, as a function, and then use a multivariate function linear model for the analysis image RNA-Seq data. So that's some mathematical, okay, because I am a statistician, I should give some, but not in detail, okay? So, in fact, this is the XST, for example, for image signal, the location, if they can be two-dimensional or three-dimensional. For simplicity, I just give the formula for two-dimensional. However, this one can be easily extended to the three-dimensional. The location, they have two locations. And this is just a weight. By some statistics, we try to find the variance of this one, how to choose the weight function to make a variance of this signal largest. And then by some statistical working manipulation, we just try to maximum this integral under some condition. Finally, we can get this integral equation, and this is the beta ST, this is the principal component function, or just the weight function. And then each image, we can use this signal. This is called the function principal component score, represent some signal from image. So now this is formula. Now this is the signal from image, and then this is the function principal component. Previously we derived, and then we can calculate this one. Here I give example why I should use the function principal component analysis, try to reduce the image signal. This is the original image. We only use six function principal component score. You can drag this picture, you see? Very similar, right? However, If not previously, most people use, for example, 40-year expansion. If we use full expansion to check the image, you see, more than 60,000 components. And the picture is still not clear. You see, this one, you see, yeah, not that good or not well approximated this one. Now here I give other example, not just the six, all this use six function component score. You can check this one picture. This picture is very clear, right? And then now for CT, we study the cancer. This is normal. This is original image. This is constructed image by function component analysis. Very similar. This is the early stage of pancreatic cancer. You see, this is original image. This is the reconstructed image. This is the advanced cancer, original image, and then this is reconstructed image. So here, clear demonstrate the function component analysis work very well. So now we should, second one, I should multivariate function linear model. What do you mean? So this is the Y represent the signal from image, and here this one is just gene expression, RNA-Seq. We take RNA-Seq gene expression as the curve of the function, and the T is the genomic position, and then we use that function linear model. Now by some mathematics, this one can be finally reduced to the traditional multivariate analysis. For example, here, we traditional multivariate regression analysis. This is the Y image signal. This is the signal. This is the information from RNA-Seq data, and this is the coefficient that represent the effect, the expression on the image, and the test statistic. This is the chi-square test statistics. So now, Y position matter. This is a very simple example. You see, we have a control by treatment. On the average, gene expression is the same, right? This one. However, for this one, the second set exon, they have more expression than treatment. However, for the isomer 2, the treatment first and second exon have more gene expression in the treatment than control. So we should consider their variation. Now here is the example. Now we say, in fact, we observed data. You see, this like a function. This second one, this one. Even if you say maybe the average similar, however, the gene expression curve. This is this one are different. Here, if we cluster gene expression data, we use function component, you see the three clusters, and then the shape. Now, in each cluster, the shape almost look similar. For this one, however, if we use never reader means you see one gene expression, one number represent them. You see, even if in the one cluster, the shape not look the same. So now, this is the idea. We give some application. So first we applied to ovarian cancer data. So this data, we have total sample 176, and then drug sensitive 106, and the drug resistant 76. The image now we can they might not pick it up. This it's just that we can divide it into 16,000. Total number of genes 13,000, and then significant level p-value. We declare okay, this gene is significantly associated with the image or not. This is the number here. We identify 21st the genes are significant associated with the image. We first look at the QQ plot. That means our statistics. That means good. Okay. And then here, use our method wait, now this is the p-value. If we use other descriptor, you see, no, they're not significant, you see, summarize the image signal. If we use RNA-Seq data, if we one number represent one gene expression, almost not significant, right? The second one, just the kidney cancer. This is normal. This is tumor. Now you see, image are different, right? So here, image 188, and then cancer 121, normal 67. The number is in more than 16,000. This is the p-value for declaring significance. Here, number here, we identify 84 genes are significant associated with signal. Now, when we submit the paper, some reviews, okay, why I use the image? Why? Because image also associated with diseases, right? We're not here. Now, we use the image to classify the cancer. We get the average. Now, we use 5 cross validation. We get the average accuracy 83% is good. So also, Google plot is good. Now, we show also again here, this is our method, the p-value. This is a descriptor, p-value. And this is just take one gene, one number, one gene expression. See? The same. We can do not the same. Here, the good thing is this one. We identify the genes associated with image signal. You see, these genes form protein in the next network, and this is for the kidney. Here the splicing site from our data analysis change. Here change. The splicing site we get some picker here. We can identify splicing site. Here I say many genes are not differently expressed. However, you see from this cluster we can see many genes are splicing. We can observe splicing site here for this one, second one. So now my conclusion is this one. So using imaging and gene expression data analysis, we can identify genes which may not be differentially expressed. However, they were associated with the image may be these genes are change the shape of cell tissue. Thank you.
We do have time for one question. If anybody has a question. One question? Okay.
Few years ago, I was interested in studying leukemia induced by HDLV virus. And I know that this leukemia induces modifications in leukocytes, that is the nucleus acquire a form of flower, the flower. And I was really interested in know why they acquire this so pathognomonic shape. And at this time I was studying differential expression by suppression PCR. Of course, at that time I would not have any condition to clarify this problem. Do you have this? Do you think your approach will be for elucidate this question?
Yeah, I think maybe you needed you from needed to your observation. You see, in fact, you see, I just looked at there are 3 dimensions to structure protein. I found some spring side, how to affect 3 dimensions structure of protein. So you observe. Now I think maybe you say unrelated. Now I'm not the expert, you see, on this area. However, my feeling is that they maybe you see, now we change some shape, some protein structure. Okay, thank you.
Thanks, Dr. Xiong.